So very good afternoon, everyone. And today is Gao Zheyu's final presentation. Okay, and the title of his final present of his project is the effects of quercetin on cancer cell lines. So today with me is Supervisor Associate Professor Dr. Koo. The reviewers are Dr. Chuai Lan and Dr. Nur Shahida. And then when you are ready, you can share your slide first. So Gao, are you ready to share your slide?
Okay. Can you hear me clearly? Because
Yes, I can hear
there
you, but you
is
can speak up a little
some
bit more louder.
yeah. 怎么搞来着？
Okay, do you know how to share your slides? Could you share your slides?
Yeah, I'm sharing.
Okay, so whenever you are ready, Gao, you can start your presentation.
Okay. Thanks for doctors for attending my speech, and I'm Gao Zeyu. My supervisor is Dr. Hu. Wow. So why am I doing this research? Because the cancer problem in today's world has become increasingly serious. The World Health Organization says there will be an estimated 10 million new cases of cancer and 9.7 million diseases in 2024 globally. About 1 in 5 people will develop cancer in their lifetime. About 1 in 9 men and 1 in 12 women will die from cancer. Cancer has become the second leading cause of death worldwide, especially in economically underdeveloped areas. So we need to know the costing and necessity and the GEO. The costing is the plant flavonoid belonging to flavonoid group of polyphenols and has strong anti-cancer properties. Costing can effectively inhibit the growth and the proliferation of various cancers. Costing can also reduce the cell viability, autophagy, or promote apoptosis by regulating signaling pathways. Necessity is an in vitro drug discovery platform representing 9 tumor types, which is used to continuously screen potential driven to drugs and study drug response. GEO stores the genetic date of targeted cancer cell lines we need. For the problem statements, I found a large number of epidemiological studies highlighting the potential of costing to reduce cancer cell viability, growth, and proliferation. However, only a few cancers will focus on more common cancer cell lines that might respond positively to costing treatment have yet to be elucidated. As such, preliminary study aims to investigate the effect of costing on 60 different human cell lines using the information in the CST database to identify and characterize different cell growth inhibitory effects upon costing treatment. About the research question, can the CST cancer cell line panel provide significant information for our study to select the best cell line upon costing treatment? What gene expression fields are shown in the GO of the selected cancer cell line upon costing treatment? What are the levels of the growth inhibitory effect of the costing-rich subtraction of renin phenolic crude extract on the selected cancer cell lines compared to pure costing? What are the levels of the Ki67, PCNA, and the PGP gene expression in the costing-rich subtraction of red onion pure ethanolic crude extract on the selected cancer cell line compared to pure costing? My hypothesis, this study hypothesis, this hypothesis and approach where a cell line or field is identified from the selected cancer cell line panel, which displays the best cell line upon costing treatment. When browsing the GO response, the gene expression profile associated with the growth inhibitory effect of costing on the cell line can be analyzed. The growth inhibitory effect and field gene expression, Ki67, BXCA, and P-glycoprotein of the costing-rich fraction of red NPO ethanolic crude extract compared to pure costing can be explored on cell lines by cell-based proliferation assay, offering a fresh perspective on cancer research. My main objective is to comprehensively identify the growth inhibitory effects and its related genes expression of costing on cancer cell lines. Divided to three specific objectives to identify the cell line that shows the best growth inhibitory effect on costing treatment in CST cancer cell line panel, to analyze the gene expression associated with the growth inhibitory effect of selected cell lines in GO response theory, to investigate the growth inhibitory effect and field gene expression upon costing treatment and upon treatment with costing which the fraction of red onion pure ethanolic crude extract compared to pure costing in the selected cell lines by cell-based proliferation assay like LDH. To achieve all the objectives mentioned just now, I first lost the NCST and GEO. The NCST database was first accessed through the NCI DTP to select the cell lines showing effectiveness upon costing treatment. High throughput functional genomic data of selected cell lines were studied through the GEO database of SBI to annotate gene identity, dataset title, links to supporting information, and growth describing the expression level and rank of the gene in each sample in the dataset were included and stated. After that, affected cell lines will be controlled and treated. A cell will be treated with the cotin-rich subfraction of radium pure ethanolic crude extract, and for comparison, pure cotin will also be used to treat this cell. After this treatment, the his cell will be subjected to the anti-proliferation assay, like LDH. The cell morphology after the treatment will be also be monitored under a microscope. Treated cell will also undergo totally RNA extraction and cDNA synthesis. The cDNAs will be used to detect the identified genes from GO by semi-quantitative PCR. Finally, GutPad Prism software will be used to make all plots and statistical calculations. The data will be presented as the means standard deviation or at least two independent experiments. The p-value minus 0.005 will be considered as a statistical significant difference. This is a geo resource, and this figure shows the dose response curves of costin against the panel of CST cell lines. Most of the cells exhibit the growth inhibition effect upon costin treatment. And I managed to select some cell lines that displayed the promising growth inhibition upon costin treatment. Yeah, there are eight cell lines. Next, based on the GF50 values among the selected cell lines, only the RFCEM, this cell line is sensitive, but KI67, okay, 562, NCIH 522, and A 498 are resistant to the costin treatment. SF 539 and U251 showed no effect on treatment, and no information was found on the this MDA MD 435 and the HS 578T cell line. So we select the 4 cell lines, and finally we choose the K 562 to do the LDH. I see. Based on the dataset submitted to the GEO database for project intensity, costing effect on gene expression in HepG2 hepatocellular cancer cells. The dataset was divided into the two groups, one treated with the costing and the other serving as the control group. Treatments are applied for 12 and 25 hours. This is phenomenon of differentially expressed genes are observed in the selected genes in this heat map. The graph will represent the difference in gene expression, specifically the observance level of three selected genes, KI 67, PCNA, and the people glycoprotein under these conditions. Log for the changes was used to quantify the magnitude of gene expression changes. During treatment and control conditions was showed the various gene expression levels over treatment period. Positive values indicate the upregulation, mean the gene expression in this in the treatment group is higher than the in the control group. Negative values indicate downregulation, meaning the gene expression in the treatment group is lower than the in the control group. This phenomenon allows for a quick assessment of how target gene expression is affected by treatment log plot change is typically used in combination with statistical significance testing, such as the p-value and just p-value. P-value indicates whether that changes is statistically significant. Gene is generally considered significant differently expressed when the absolute value of log plot change is greater than center threshold, and the p-value is less than 0.05. In that case, the mRNA expression of Ki67, PCNA, and P-glycoprotein genes is not statistically significant under the treatment condition for the dataset. There are four different different figures. We can obviously see the costing exhibit the almost no effects on the transcriptional profile of HepG2 cells. Yeah, this is HepG2 GEO results. And then I use the LDHC to detect the K562. The principle is the more the cell are damaged, well, we can see the more the higher absorbance origin. So I get after I use the LDHC to detect the two cell lines K562 and HepG2, I get this 4 figures. As shown in these figures, and little or no detected detectable LDH were released from K562 cells after costing treatment at various concentration for 24 hours. All curves were indicated below the Y axis is zero line. Similarly, K562 cells treated with subtraction of non-pure ethanolic extract for 2 hours and 4 hours also showed little or no detectable LDH release. The optimal cytotoxic effect was only observed after 24 hours of subtraction treatment with the EC50 of approximately 34.4 milliliters and 7.5 microgram milliliter at 12 hours and 24 hours respectively. Whereby the idle dose response curves of subtraction showed high potency at 24 it was then at 12 hours of treatment. On the other hand, HepG2 cells treated with costin and the renal pure ethanolic extract subtraction showed little or no detectable LDH release. The curves of ideal dose response curve were only observed at 12 hours or 64 hours of subtraction treatments. The optimal cytotoxic effect of subtraction treatment was observed to yield EC50 of approximately 69 microgram microliter and 24.5 microgram per microliter at 12 hours and 24 hours, respectively. This part of the study indicates that both cell lines are sensitive to treatment with costin-rich compound, despite the subtraction effect being more potent in Ki-62 cells than in HepG2 cells. This PCR results is only for 562 cells. For mRNA expression of Ki67 pCNA and B glycoprotein, cell analysis did not detect any significant effects of Ki67 in control of subtraction treated K567, K562 cells. However, PCR analysis detects a significant reduction in pDNA mRNA expression in subtraction treated cells after 24 hours compared with control K 562 cells. This phenomenon is consistent with the growth inhibition observed in K 62 cells using the LDHC after subtraction treatment. Similarly, PGP a gene associated with the drug resistance also showed reduced gene expression in the subtraction treated K52 cells collected at 24 hours of treatment. My further work will further demonstrated the growth inhibitory and cell cycle effect of costing rich subtraction in K52 cells using assays like such as CCK8 and anything way 3D spheroids may also be used to assess the subtraction effects on K562 cell structure and microenvironment. It's my guarantee. Okay, thanks for listening.
Okay, thank you, Gao.
There is so noise because my neighborhood is doing the renovation.
Okay, thank you, Gao. So with that, we would like to invite either Dr. Chu or Dr. Shaida to give comments to Gao about, or ask questions about his presentation.
Dr. Chu, you want to start first?
Can you hear me?
Yes, can hear you.
Yeah.
So, Rashida, I start first, okay? Is it okay?
Yeah, okay.
Okay, Gao. Yeah, thanks for the presentation. My first question would be on slide 3, right? You said the effect of quercetin on cancer cell lines is well documented but not well studied. Yeah, the last line, well documented but not well studied. What do you mean by that?
It means this tool is very good, but the study is not enough.
You mean the data is there?
Huh?
Can you repeat your answer again?
This test is just mean I think this tool, CFT platform, this tool is very good, and but there has also many concerns we didn't use this tool to study.
Oh, okay.
Just this.
Okay. So another comment is I think all your slides you use future tense, right, in your methodology and all that. I think you should change them all to past tense. Okay, especially for your methodology. Okay. So shall we go to slide 17?
17.
Yeah. Yeah, I
Oh,
just wonder,
long.
your slide 17 and 18, all that, right? How come there is no separate results for your two cell lines? You have two cell lines, right?
Oh, no. This is just happening to sound. This, yeah. This geo result is just the happy two, because it's from the geo the website.
So this one is for
5G2.
HEPG2.
Yeah.
How about your K562?
No, because K562 is I selected from this one. Is 67 is the set, and from Geo I find this and this just a sample. This HEPG2.
Meaning all your GEO is only on HEPG2.
Yeah.
Okay. Then how about, because after this I lost your slide number because I don't see any slide number on further slides. So go to slide about cytotoxicity. Yeah. So your A is pure quercetin, is it?
Sorry, what?
Your slide, your graph A
Yeah.
refers to your control pure quercetin, right?
This is costing.
Quercetin, yes. Then B
Yeah.
is subfraction.
Yeah.
So which one is your control? You compare your sample with the control, right?
Yeah. 这个是等一下。Give me some minutes. I paused many things. Control.
Or maybe I continue with my questions regarding these slides, right? Zari? Okay.
I remember something. No control.
No control.
Because the results the control have been my 叫什么？就是 minus have been minus to make this because for example we get the control data and get the costing data, and we need use the costing data minus the control data, so then we get the real data to make the chart. So what
So both these A and B are your samples.
of course
You
my
mean-
samples.
Well, A and B are both your onion extracts.
A and B,
Yeah, your graph A and graph B
graph A and graph B.
are both, because one is subfraction, one is quotient.
Yeah.
So both are your onion extracts.
I extract.
No,
Yeah. The
just
content
that
is
I just am a bit maybe
yeah.
I don't get it.
Both my
What's the
extract.
difference between your quotient and your subfraction?
It's pure costing. It's storing the I forgot some yeah one type of compound. Yeah. So it's pure costing, and the separation is red and pure extract.
Subfraction is your extract. Quotient is the one
Pure costing. Pure.
pure quotient. Okay. Then another question on this slide is, just now your GEO, right? You have only had G2, right? Then here in cytotoxicity, you have K562. But you don't have result on K562 before this, right? Your previous one on GEO, you don't show the K562, right? You show only your G2.
Yeah.
Then how come after that you have your K562?
What?
I mean, previous slides you have your result on GEO, right? I maybe
Yeah.
I need some further explanation because I'm confused about the slides as I'm not familiar with the graph. So just now you mentioned you don't have K562, you have only had G2, right?
Yeah.
GEO. Then how come the cytotoxicity? Then you again you bring up K562.
Actually, how to speak, my plan is use NCICT the platform to select one the most the costing resistant the most costing resistant cell lines. So I select KK562 because only the pure costing resistant we can find out the subtraction random impure ethnologic extract is its effect is for is or not for real. And The FG2 is actually as for comparison, we can because it's started by some people, and we can briefly see the HAPG2 cells is of course the Pro is not enough, but we can also see the HAPG2 is have some resistant to the hosting. So
Okay.
uh,
Yeah. Okay. Then how do you interpret your graph A? Meaning the quercetin has no, because they have no cytotoxicity activity against your cell line, is it?
what?
Graph A, right? Because all
Yeah.
the data are below 0, right? Meaning no cytotoxicity against the cell lines.
Yeah.
Okay. Then I just wonder, like, why the axis S is your concentration and not the time? Like 2 hours, 4 hours, 12 hours, 24 hours. Why do you choose to put it that way? Your axis X, instead of putting time, you put concentration. Because to me, it would be easier to, I don't know, like, normally it would be easier to see the effect, the time base, right? Why do you choose to put it this way? Like, to me, if let's say your axis X is time, right? 2 hours, 4 hours, 12 hours, 24 hours. So you can see the changes time base,
Yeah.
the trend when the time change, then how the thing go accordingly to the time, right?
Yeah.
How it develops along the time. So how come, why you put your concentration as a axis X instead of the time?
I don't know what is axis. Axis sharp.
X, because your graph you have your axis X and Y, right?
Yeah, axis.
Axis X is concentration versus cytotoxicity, right? So your concentration, why don't you put the time? Because time, you see, you have 2 hours, 4 hours, 12 hours, 24 hours. So how if you put your hours as your axis X, then we can see the changes over time, right? Your observation over time, the results over time, you can see the trend. Then your concentration, you have different graph or the line for different concentration. You see what I mean? Because if in this case I cannot see, I don't I'm a bit confused myself. I don't know.
You want to see the different R's? You can see yeah, you should different R's this one, and
yeah. Why do you put it this way? Why do you put the concentration there and not time? Because
the one reason I just can thinking is that we can see the 50. we can see the 50 value.
Because if you present concentration, right, bar chart,
Can-
right? You use bar chart, you use line graph. Then we try to interpret the line because we want to see the changes over time, right? The trend, show the trend over time. But now it's not, it's concentration. So when I read the graph, I'm a bit like, I have difficulties to interpret the graph. You understand what I mean?
Yeah.
Because the concentration is not appropriately represented in line graph. So to me, it would be more, if I, as a reader, when I read this graph, I would expect the axis to be time based, so that we can see the trend, right, over time. You understand what I mean?
Yeah. I mean,
So I just would like to know what is the logic that you present your graph in this way. Maybe you have your justification to present it this way, right? I might not know because I'm not the expert doing this work. You are the one who do and who present. So you should have your justification why you want to present this way.
I'm never thinking this.
I'm not saying that you are wrong or what. I just want to see
Oh,
why you do this. I just try to understand what is the logic of it.
Because it's more short. The if we want to find a medicine, and this one, this figure maybe more clearly to how yeah, the concentration concentration of the medicine. Uh, yeah, I never think this.
Okay, you go to next slides on gene expression. Yeah, all this graph I think actually you should separate them because I cannot see, like you have the T graph on your right hand side, right?
What? Can I say what?
Yeah. My advice is for you to separate the because now I cannot see what is on the axis. I have problem with the axis X again. What is on the axis X here? The tree graph, colorful graph. I cannot see them because to me it's too small for me to read. What is on that slide?
Yeah, this one. Tree.
The X, axis X. What does it mean? I cannot read it here on my screen. The blue, what is there's a blue bar stand for? Yeah. Yeah. What is that?
This is
What is blue? What is green?
What do you mean?
Just
Wait, what do you mean, little?
sorry, because my screen, from the screen I cannot read the thing. What is blue? What is green? What is orange? What is purple?
From this, this is this tool. This tool is the
No. I'm referring to the colorful graph, right hand side one.
colorful.
Yeah. You have graph, right? The bar chart.
It's same. It's same. You can see the color. It's same. This one is this one. This one is this one.
Oh, your control. Okay. I'll just translate the image into.
This is control 12. This is subtraction 12. This is the 5 and the 6 is control 20. Sorry. Control 24, and the 7 and the 8 is subtractions the 24.
Okay.
Because it's different with this order, so I get this.
Yeah. Okay. I think I shall stop here. Thank you, Zoe.
Yeah, thank you, Dr. Chu. Okay, and Dr. Shaida.
Okay, thank you, Dr. Eugene. Okay, Zayu,
Yeah.
can you show me the slide on, do you have on objective? Okay. All right then. I think in your last slide, you should have one slide on summary and justify whether you have achieved all these objectives. I didn't see any slide on summary because what you put is future work. But to be frank, it is hard for me to understand your work. I have to refer to your proposal and read again. And your answer to Dr. Chiu's question, I think you didn't really answer. I'm not happy with or the way you answer because no, there are some missing points that you didn't really justify why you choose HEP 52 K562. Do you understand?
Not really understand.
Not really. Okay, so maybe I go one by one. So first, to help us better understand your work, maybe in your slide you should put what is the main point that you want to deliver. Like for example, just now the toxicity slide. Okay. You want to compare the two cell lines, right? Okay, so you just write at the bottom of this graph, you just write whatever you want to say, because the way you present, we couldn't understand, we couldn't really see what is your point. Okay, now you have the quercetin, but just now when Dr. Chiu asked you whether this quercetin is your control, you said there's no control. But then you say, okay, this is your quercetin you take from vials and do it. That means it's a control because it is not that you produce the subfraction. So just write at the bottom what the point that you want to compare between the subfraction and the quercetin, the findings, okay? And then I think you need to be consistent. When you put quercetin, you just put pure quercetin. That
Yeah.
really refers to your control, the one that you put in your proposal. Because when you put quercetin and then you mention the pure quercetin in your proposal, that some kind mix up. And then when we ask you whether it is your control, then you mention it's not your control. Okay, to help you better understand your work, you just write whatever you want to explain about this graph at the bottom of the graph, then you put your findings, okay? So I don't, whatever questions by Dr. Chiu is actually the one that I want to ask you, but I'm not really get the answer from you when you answer to Dr. Chiu. Yeah, maybe I just repeat again so that, do you have a pen with you right now? Can you please jot down, like, just now Dr. Chiu mentioned about the slide number.
I can get this recording. Yeah, no problem.
Recording. Okay, so first is the slide number. It is not consistently.
Oh, yeah.
Okay. And then my suggestion is to write your findings, the points that you want to explain when in every slide, so that we better understand what you want to say. Your past tense to your methodology, write everything in past tense because you have done it. Add a few slides more on summary, a limitation of your work, then only you put your future work. Okay?
Yeah. Yeah.
So that we can see the conclusion. What have you got? What is the highlights that you want to tell people about your work? Just now I asked about the objective. I just want to see whether you have completed all your work. So do you have any more work left to be done, or you have completed everything?
Yeah.
Okay. So okay, then maybe then you have your summary lah. All right. About the selection of the two cell lines, the K562 and HEPG2. Okay, in your table, you put 562, the resistance cell lines, but on the HEPG2 is based on the geo, right?
Yeah.
I hope you put it in your justification, in your thesis. Why is that so? Because we want to see these two in the table, perhaps, or in the geo, something that the selection is not biased. It's not based on different character, different. It is based on same definition, something, right? On the, can I see your bar graph just now, the colorful bar graph? Slide 26. Okay, this one, your comparison in your slide compared to your proposal is not the same. You mentioned in your proposal is 72 to 96 hours. But here you just like put it on 12 and 24 hours. Why is that so? Do you change your parameters or what?
Because from this one we find the subtraction in 12 hours and 24 hours it have effect, and so we use, because in 2 and 4 hours we don't find the effect.
To find the effect.
Yeah, so of course we use the effective.
Okay, when it's actually when you compare between this expression K167, 12 hours and 24 hours subtraction, and the control is quercetin, right? The green, you compare the green and the blue. The red and the yellow.
Red and yellow. What is yellow? This
orange. You mean orange?
Orange. Yeah.
So what you mean there's no difference?
Red and orange is no difference. You mean this?
I'm asking you, is there any difference because you put the bar chart, you put the standard deviation there? Do you have the p-value?
No, I don't have the p-value.
If you don't have the p-value, then how you do the standard deviation?
Oh, I don't show the p-value. I don't.
So you have the p-value or you don't have the p-value?
I have, but yeah. I have, but it's maybe it's in it's stored by me. 这两个。对，
This bar chart, do you plot it based on what data, what value? Because on the left hand side you do gene expression and you do.
Actually it's this one. Maybe you can't see, but actually it exists. This one. It's so not so not sharp. So I think the KX7 is the outcome is not very good. And
Okay. Now the band on
yeah.
the left hand side, the band, you run the control 12 hours, control 24 hours, and then you compare with subfraction 12 hours. So you can see there's only 3 bands on the subfraction lane. Okay, you didn't see any bands on, if you refer to KI167, on the left hand side, there are 4 bands, the right side, 3 bands. There's one missing on the lane subfraction 12 hours, right?
You mean this?
The upper one, the top one.
Oh, yeah.
Okay. Then on the bar chart, the green one, the bar chart is so high that, it refers to the subfraction 12 hours, right? The one that you can't see any band on the...
This band is arrow.
Yes.
Yeah.
So what does it mean?
What does it? It's not mean anything, ma.
Sorry?
It
doesn't mean anything.
It doesn't mean anything, ma, because it's arrow. Arrow can mean what?
Okay, I don't really understand actually. Takpelah. Okay. Yeah, I think I have highlighted few issues, but to me, you have come a long way. You should be able to present. Your slide should be ready with slide number, conclusion, something like. Now you have presented so many before, so you don't have to rely on your supervisor to check on your slide, because you should be able to have that skill.
Yeah.
Okay, so just be careful on the small meticulous on the slide number, the point, the highlights, the summary, so that people can easily understand your work. Not right. Okay. Yeah, that's from me, Dr. Eugene.
Sorry, Dr. Eugene.
Yeah,
I have
okay.
one last question for Jerry.
Sure.
For these slides, right? For these slides, your blue and your orange charts, are they replicates? Are they both? No, these slides, right? Your bar, your blue chart and your orange bar, right? The blue bar and the orange bar, are they replicate? Are they replicate because it's all both
No.
Control 12 hours.
Not replicated. What?
Oh, different gene. I cannot see your axis X. That's why I'm confused. No, control 12 hours.
Zhe, see how I answer the questions. They are from different experiments. The data were obtained from different experiments. Okay.
Oh.
So later, I will display the recording again for you to have your practicing, more practice. You need more practice.
Oh, okay. Now I see. It's round 1 and round 2. Okay. Okay, I think he has also to improve the slides for presentation, and in future during the Viva, I think maybe he has to separate them out or something so that you can have bigger. Yeah. Or else, you won't be able to see it. Okay, we don't have we don't have the copy with us. Okay, thank you. Thank you. That's all from me, Dr. Eugene. Thank you.
Okay, Dr. Chiu. So Dr. Koo, do you have some other comments?
I don't have specific comments lah.
Yeah, okay. All right.
Because the students I think should have more practice.
Okay, yeah.
Yeah.
So yeah, Gao, I think
So
you
thank.
have improved a lot already, but still there are some
Yes.
comments where you can make further improvement.
I think the improvement I can see on the confidence to answer questions.
Yeah. Okay, so one suggestion that I can give you is to add a one-line summary for your slide, Gao.
What?
Yes, because you are not English speaker, so usually for us who are not English speakers, we can add one summary in each slide. For example, this data slide, you can add a small summary in maybe one or two sentences just to help explain what is this slide.
Yeah.
Yeah, so you
And
can
also,
add.
do you have somebody sitting beside you, or you still rely on the translator? Can you turn on your camera?
Camera. Camera 是啥？
Camera, yes.
Yeah, for the dissertation presentation, you have to turn on your camera. For
Because
today's presentation, it's okay, but for the
I
Viva,
heard another voice from when you are presenting. You were presenting. So try not to use translator, otherwise you can never improve your
没
English.
用啊。
Don't speak other language.
it's
With me,
okay.
you know.
I think Gao is still trying his best. Yeah, that's good.
But
So
yeah, if I have the time, I will let him practice again. Anyways, send the recording to me. Yes, as usual I will let him listen back what he presented just now.
yeah, okay, yes. So later we can get from Mr. Anas
Into
for the
Anna's slide.
recording. Yeah.
Okay, right.
Okay. So the dissertation final Viva will be after I think
December.
after December, January, I think. Yeah.
December?
No, you will submit your thesis in December, right? So your Viva is usually in the January.
January
Yeah. Okay.
这样 OK
Yeah. So you can continue to practice and improve and take up. And later, I think Dr. Chiu and Dr. Shaida will give the written comments as well to help you improve on your slides and the data presentation. Okay. All right. So thank you, Gao, for your presentation. And
Thank you, Eugene.
yeah. So thank you, everyone. Dr. Chiu, Dr. Shaida as well.
Right. Yes.
Yeah.
Thank you, the evaluators, Dr. Chiu and Dr. Shaida, for your comments.
All right. Okay. So we will
And
end.
also
you,
Dr.
Esther.
Eugene for chairing these sessions. Thank you so much.
Yeah, no problem. Okay.
You.
So, see you again.
Okay.
Bye.
See you. Bye.
Bye-bye.
Bye. Hey. 是什么